Okay, let's get started with chapter 3. What I'm going to do is insert YouTube video clips throughout the PowerPoint presentation so that you actually can see things, because I know that everyone learns in different ways, so rather than just have you listen to me speak, which can be incredibly boring after a while, I'm trying to inject some different learning methodologies as best I can in this kind of online environment. So there will be a few YouTube clips coming along. So what we're looking at with chapter 3 is biological foundations. We're looking at understanding genetic inheritance, the transmission of genetic differences, how genetic abnormalities occur and what response can be made, and also the joint influence of genetics and environment. Now, you're not having to study the entire chapter. This book is incredibly dense and I'm only getting you to look at certain sections, so make sure you pay attention to the chapters or to the parts of the chapter that we're looking at and that I'm requiring you to understand. So here we go, the mechanisms of genetic transmission. Now, some of you who've studied biology will remember this or not, and those that haven't studied biology, this may be the first time you actually come across this, so I'll try and keep it as simple and as clear as possible. So the gametes, these are the reproductive cells of your parents. In the father, gametes are produced in the testicles, and each one is called a sperm. In the mother, they develop in the ovaries, and each one is called an ovum or an egg. The sperm and the egg cells contain genetic information in molecular structures, and these are called genes, which in fact form threads called chromosomes. And the chromosomes contain the genetic material that you as a child inherit from your parents. And each human sperm or egg contains 23 chromosomes. All other cells of the body contain 46 chromosomes. So this diagram that you can see here shows the karyotype of chromosomes for the typical human male. And you can see that's an X and a Y. The final two chromosomes are the sex chromosomes, as in this example, that's a male and it's an X and a Y. If it was a male, it would be an XX pair. So I've included here a couple of very cute little clips which make this whole process of understanding what genes are quite easy. They're pretty quick clips, they're only about 3 or 4 minutes each. I would recommend that you watch them, so you can just pause for the moment and watch these and then come back and listen to the rest of the lecture. So now we're on to the role of DNA. DNA, commonly known as deoxyribonucleic acid, it is basically what genes are made up of. They were discovered by Watson, Crick and Wilkins in 1953. It's transformed the way we know everything about humans. Now, when a sperm penetrates an egg, they release their chromosomes, which join together to form a new cell called a zygote. These cells then make copies of themselves through a process of meiosis. And meiosis is this process where the cells, or the gametes, divide and recombine into a zygote at conception. All other cells that make up a unique human being will develop from this original zygote through divisions of their genes, chromosomes, and other cellular parts through a process of mitosis. And here's an example of mitosis. So this process ensures that the duplicate cell is identical in genetic makeup to the original cell. Chromosomes copy themselves, and therefore each new body cell contains the same number of chromosomes and the identical genetic information. So meiosis is the cell division process where the gametes are formed. So that is a process of reduction and division, which halves the number of chromosomes normally present in body cells. So there's actually a number of steps that happen. The 23 chromosomes of the egg or the sperm duplicate themselves, and they break up into small pieces and randomly exchange segments of genetic material with one another in a process called crossing over, in which genetic material is exchanged between pairs of matching chromosomes, one from each parent. This creates hereditary combinations and recombinations. There are millions of possible combinations, and the chromosomes divide to form two separate cells. Finally, the two cells divide again, and each of the 4 new cells contain a unique set of genetic material in its 23 chromosomes, 1/2 of the usual number of chromosomes carried by all other cells. And this ensures that the new single zygote that forms during conception will contain the normal 46 chromosomes. That is 23 from the egg and 23 from the sperm. And this figure that you're looking at now illustrates the process of meiosis for sperm cells. And basically the process for egg cells is the same. Now you don't need to, I'm not requiring that you know and understand this process. It's really just about knowing that the process occurs, and this is where our DNA and how our cells are created, so that we can see what happens further down the track when how we develop genetically. So we have these two things called genotype and phenotype, and the genetic information contained in our cells influences the development of our unique physical, intellectual, social and emotional characteristics. So the genotype is the genetic information inherited that has the potential to influence observable characteristics, that is, eye colour and height. So these are the things that we can observe. The phenotype are the physical and behavioural traits an individual actually exhibits, that is, blue eyes and the actual height. So the product of the interactions of an individual's genotype and environmental influence creates the phenotype. So in some cases we see a close match between genotype and phenotype. So for example, inheriting genes for blue eyes generally results in actually having blue eyes. So in other cases they may not coincide so closely. For example, weight is influenced by environmental issues of diet. So the genotype is made up of those inherited traits. So how to remember this? So the word gene is part of the word genotype. So these are your inherited traits. The phenotype is what's actually expressed. So this is the fact that you might have curly hair, or you might have blue eyes, or you might be 6'5". That's your phenotype. So that's how the gene expresses itself. Here's a good little YouTube clip on phenotypes, and this will help you to remember this one a little bit more clearly. So the genetic expression. We're looking here at the, and we know that genes are inherited in pairs, one from each parent. Some genes are dominant and some genes are recessive. So a dominant gene will influence a child's phenotype, even if it is paired with a recessive gene. So remember, the phenotype is how the gene is expressed, how it looks. A recessive gene must be paired with another recessive gene to be able to influence the phenotype. If it's paired with a dominant gene, its influence will be controlled or blocked. So there are more than 1,000 human characteristics that appear to follow the dominant recessive pattern of inheritance. Eye colour is an example, and this next slide really shows you how that is an example. So the determination of sex, how is it that whether a person becomes male or female depends on various events at conception. So all ova, or egg cells, contain a single X chromosome, whereas a sperm cell may contain either an X or a Y. So if a Y-bearing sperm fertilizes the egg, a male or an XY zygote develops. If the sperm is X-bearing, a female XX zygote develops. So there are more Y sperm than X sperm that succeed in fertilizing the ovum, which results in about 30% more males than female zygotes. At birth, male babies only outnumber females by around 6%, and by 35 years, women begin to outnumber men, suggesting that males may be more genetically vulnerable than females. So vulnerability is actually linked to sex-linked transmission because males have XY and females have XX. So the Y chromosome is shorter than the X and therefore may lack many of the gene locations of that X chromosome. Many genes from the mother may not be matched or counteracted with equivalent genetic material from the father, and as a result, genetic abnormalities of the single complete X chromosome are more likely to result in phenotype abnormalities in males than females. So examples of sex-linked recessive traits include colour blindness, which is usually red-green, haemophilia, which is the inability for your blood to clot, or muscular dystrophy, which is weakening and wasting away of muscles. So once again, another little useful short YouTube clip, where do your genes come from? Which I'll give you a couple of minutes to have a look at. So on to genetic abnormalities. There are many, but these are probably some of the ones that may be a little more mainstream than others. So the chromosome abnormalities, and the example here is Down syndrome. Persons with Down syndrome have 3 number 21 chromosomes instead of the typical 2. Specific phenotype of member phenotype is how the gene is expressed. So the specific phenotype of almond-shaped eyes, round heads, stubby hands and feet, and many also have heart and intestinal tract abnormalities. They've also got a greater risk of leukemia. Most live until middle adulthood, and they're more common in babies where the mothers are over 35 and there are older fathers. Huntington's disease is a dominant gene disorder. It requires only one abnormal gene from either parent to affect the child, and it results in a gradual deterioration of the central nervous system, which causes uncontrollable movements and mental deterioration. Typically it appears in 30s and 40s and always proves fatal. Very, very sad disease. Ongoing research in identifying the specific genetic marker and the development of genetic blood tests so an individual would know before their 30s and 40s is underway at the moment. Recessive gene disorders. This is a sickle cell, or commonly known, or one of them is the sickle cell disease. It's a recessive gene disorder which needs a pair, so one from each parent, and it only affects African Americans, 1 in 635, and 1 in 10 are carriers. And what happens is the oxygen carrying protein in red blood cells takes on an abnormal rigid shape inside the cell, and that gets caught in the blood vessel, which cuts off circulation, reducing oxygen supply and causing pain. And the majority will die before 20, and few live past 40 years. There is no cure yet, and they are trying to develop some treatment. And the last one, which is the multifactorial genetic abnormality, is congenital heart disease. It's a structural and/or electrical abnormality of the heart, and it may respond to medication or corrective surgery, which is performed after birth. It affects about 1 in 125 babies, and it results from a combination of genetic and environmental factors. The incidence of multifactorial disorders varies widely in different parts of the world, largely because of the great differences in existing environmental conditions. So there are ways to actually diagnose for genetic disorders, and I'm not going to read it, you can have a look at it, it's also in the text, but there are many ways now to diagnose before babies are actually born, whether there is any kind of genetic abnormality happening. So the nature versus nurture interaction is actually a very interesting one, and we will come back to this many times throughout lifespan development because it's quite useful. So behaviour genetics, which is this untangling of the effects of hereditary, which is nature, from the environment, which is nurture. So behaviour genetics is the scientific study of how genetic inheritance, which is the genotype, and the environment experience jointly influence physical and behavioural development, which is the phenotype, so how that gene is expressed. So there are key concepts inside behaviour genetics. So there's a thing called range of reaction. And this is the range of possible phenotypes on an individual with a particular genotype, which they may exhibit in response to environmental contexts. So if you're born with different genotypes for intelligence, the outcome may differ depending on environmental input. So it's the importance of an enriched environment. There's also a whole series of what we call adoption studies. So family members share environment but not the genetic background, or they share genetics but not the environment. So these studies can help to show the influence of each part. However, there are issues concerning adopted children being particularly wanted, which may influence findings. So also adoptive families likely to have been carefully matched for social, economic status, religious beliefs, cultural, etc., which raises questions as to how different the environments really are. Then there's twin studies, so they compare identical and fraternal twins raised in the same family, or twin adoption studies where identical twins are raised apart since birth, but that's very rare. Then there's also linkages and association studies, which allow researchers to identify things called polymorphisms, certain segments of human DNA that are inherited in a predictable pattern as genetic markers for the gene near where they are located. So linkage studies seek polymorphisms that are co-inherited. Associated studies compare the relative frequency of polymorphisms in two populations, one with a trait and one without. So interesting to continue to see this as we go through the various lifespans. What's really interesting is there was an experiment done around nature versus nurture, and this is a great little YouTube clip, goes for about 5 minutes. I would suggest that you have a look at it because it is quite eye-opening, and many students really find this one very interesting. And then finally, we were going to talk about in the tutorial about gene doping. Very, very current topic at the moment, particularly with the Olympics about to start. So if you want to take the time to have a look at those, go right ahead. We'll talk about it very briefly in the next tutorial, but we don't have a lot of time to spend on it because of the fact that we're missing that one this week. So feel free to take a look at these, some interesting things, and I'll also post a couple of very recent articles on gene doping that we were going to talk about, but are quite interesting in relation to sport. So that's it for chapter 3. I'll be back shortly with chapter 4.